Good afternoon, everyone. We will be getting started in just a minute. All right, let's get started. Good afternoon, everyone. Welcome to Bite Size Science. My name is Ana Sangronis. I'm the Florida Sea Grant Extension Agent in Miami-Dade County. And thank you for joining us for today's episode, which will feature Dr. Vincent Encomio, who will be speaking about the Phytoplankton Monitoring Network. This is the final webinar in our Citizen Science series for 2022. This webinar will be recorded, so you'll be able to access it after the fact and share it with your family and friends. And we just want to take a moment and mention that we hope that everyone around the state are safe and doing okay following Hurricane Ian last week. So you can use the chat box to put in questions you might have for Vincent, and he'll be taking questions at the end of the webinar. Vincent, take it away.
All right. Well, thanks everybody. And as Anna said, hope everybody is safe, and thanks for taking the time out to come today. I know a lot of people might have some Hurricane Ian related responsibilities, but if he couldn't make it, hopefully this is being recorded and you can watch it later. So what I'd like to talk to you today about is going along the theme of our webinars. Let's see if I got this here. There we go. Is to talk to you about a national program called the NOAA Phytoplankton Monitoring Network. And it's probably somewhat obvious, but I just sort of copied and pasted my name here. This is actually a slide for the program. And the real people behind this program are the two names right here, Jennifer Moucher and Nia Renee. And they are the coordinators for the NOAA Phytoplankton Monitoring Network, or the PMN. And so I've been working with them to serve as kind of a local coordinator in the counties I serve in Martin and St. Lucie. So, but they're really the ones who provide the training and get everybody set up with the materials. If you are a volunteer, or if you're interested in bringing this program to your area, whether you're at a nonprofit, an agency, or a school, those are the two people that you're going to want to reach out to, and I will show their emails at the end of the presentation. Or maybe somewhere along the way I can paste in the chat. So I just want to get into a little bit of the reasons why I've been involved in this program as a Sea Grant agent. And a big part of it is where I am in Martin and St. Lucie counties along the Treasure Coast. Some of you here may live along the Indian River Lagoon, and we're really known, its claim to fame for the Indian River Lagoon that it is the most biodiverse estuary in North America, with over 4,300 species of animals and plants. But at the same time, we have these severe water quality impacts at its base, either from freshwater discharges from Lake Okeechobee, and also watershed runoff into the lagoon and its tributaries. So this is just going back to 2018, and also I started working for Sea Grant in the end of August 2018. So I was already here and seeing these events unfold. And this is these kinds of events, as you can see here, are the satellite images of blue-green algae or cyanobacteria blooms in Lake Okeechobee during that particular summer. Red meaning very high concentrations, and bad. So you can see by the end of June, the majority of the lake was covered with pretty high concentrations of these cyanobacteria blooms. These are single-celled, really bacteria, but photosynthesizing bacteria. And that is a problem, and I'll get into that in a little bit. But that particular, the Lake Okeechobee is actually connected, and I'm going to show it here if you could see it. But those two structures that I've just highlighted, the S308 and the S80, are both navigational locks, but also water control structures that allow water from the lake to get into that canal in between, the C44 canal, that eventually connects to a major tributary of the Indian River Lagoon known as the St. Lucie Estuary. And if you look at that picture on the bottom right, that's what the water looks like when we have heavy discharges from Lake Okeechobee. So a very severe water quality impact. And 2018 was pretty unique, not just for our side of Florida, but throughout Florida. And I'll get to that in just a little bit. But with those blooms, those cyanobacterial blooms, when the water was released from the lake, ultimately through the C44 canal and into the St. Lucie Estuary, we would get severe cyanobacterial blooms or blue-green algae, as you see in the next picture on the bottom right. And of course, whenever we have those events, the health department issues advisories at particular points that they've been monitoring for contact with the water. And to give you an idea of how toxic the toxins that are produced by this particular species, in this case, it's Microcystis aeruginosa, is the main species of cyanobacteria that we tend to see when we have these blooms, and it produces this toxin called microcystin. And you can see in the red there, the concentrations there in micrograms per liter. And if you can train your eyes to, move my cursor over here, if you can train your eyes to these numbers here, these are advisory levels either from the World Health Organization or the Environmental Protection Agency for drinking water, which isn't relevant here because this is estuarine water. It's not potable, and most of the time it's brackish. But for recreational contact is 10 micrograms per liter, and you can see in the red we were way in excess of those levels in 2018. And the other thing that was really important that garnered a lot of attention locally and probably throughout the state is several dogs within a pretty short span of time, about a couple weeks. We had about 6 cases of dogs that got poisoned from contact to the water. 1 eventually died, but it was verified that they were sickened or killed from exposure to the microcystin toxin. And just to give an idea of, aside from the aspect of losing your pet, that's really, a lot of us that are pet owners, they're part of our family. It was actually pretty costly for those individuals too, with an average cost of $3,000 to try to save those animals, save those dogs, with the highest near over $10,000. So this well-documented case. I actually, just to make a little plug, one of our past bite-sized marine science seminars, I put on YouTube, and I can paste that URL in the chat at the end of the talk. But we covered a little bit this issue a little bit more in detail during that webinar. So, given that I had really just started right after all these events, one of the first things I did as an agent is, in some of the public outreach and extension activities that I was involved in, I developed a quick and dirty survey or needs assessment to determine what would be the main concerns of the public in the programs that I was reaching or engaging with the public. And not surprisingly, particularly after those events, cyanobacteria was at the top of that list. Also noted is a red tide, which was really interesting because we're on the East Coast, and red tide was really a phenomenon on the southwest coast of Florida, especially during that time. It did get the blooms of red tide did get so bad that it did start to migrate over to the East Coast, although that is not a common occurrence. But very likely the people that I had polled still had that on their minds as a water quality impact that they were concerned about. So I kind of skipped over this, but the cyanobacteria is a harmful algal bloom, or HAB. And it's we define a harmful algal bloom as an algae or single-celled algae, not just single-celled, but also some macroalgae species that can have negative impacts to humans and animals, either marine or freshwater species of algae. And these are species that are naturally occurring, but the excessive amounts or proliferation of these species may be exacerbated by activities like excess nutrients that make their way in from the watershed into these water bodies, like Lake Okeechobee that you see in the background. So, and how do we define it as harmful? They don't all produce toxins. In fact, only about 2% of all species of algae produce toxins, but they can also irritate. There's certain species of algae that have spines, and that's the one there on the top right. It's a diatom cathoceras, and those spines can irritate gills, and that can lead to fish kills. Of course, the toxins that are produced by those particular species, and some of the after effects, environmental after effects as a result of those blooms can lead to fish kills. We had a particularly bad fish kill in 2016 in the central part of the Indian River Lagoon that I'll show a little bit later. I'll get to that. But here are just some of the toxins that you can find in some of these species, and the effects that they have. Things like amnesic shellfish poisoning, ciguatera, and diuretic shellfish poisoning. These are all species that are naturally found in our waters, and they become a problem when we get to those bloom levels to the point where they're harmful. And of course, Carina brevis, for those who are joining us from the West Coast, you know that one very well. That's what causes red tide. And getting back to some of those other harmful effects, the algal blooms that had started in the Indian River lagoons that really proliferated starting about 2010, started shading a lot of the seagrass, and we saw precipitous declines in seagrass over that period, say for about 2010 to 2011, and not much recovery since then. The other thing that has happened, I mentioned in 2016, when that bloom does die off, that can create areas where oxygen really depletes. Bacteria takes over, they start consuming that oxygen, and then those low oxygen areas can be areas where fish die as well. And we had some major fish kills in 2016, which are I don't expect you to read this, but the reason why I show this slide is because it really highlighted 2018 as a very unique year where we had a very persistent red tide on the west coast of Florida. You can see by the red here the extent of the red tide bloom, and where it also even got up into parts of the panhandle, and also the cyanobacteria or blue-green algae blooms in the lake. And with those lake releases, they both went to the East Coast and also into the West Coast as well. And then the conditions were ripe during that period of time, during the middle of the summer, to sustain those blooms inherently within the St. Lucie and the Caloosahatchee estuaries. And the Indian River Lagoon is somewhat of a poster child for harmful, algal blooms, but with some harmful species. We had that brown tide that I mentioned earlier that caused a lot of the shading that eventually led to the seagrass die off. We're really still in recovery mode from that. And of course, that has manifested itself last year and this year with not having seagrass, causing a significant amount of manatee mortalities. So this is just kind of setting the stage. 2018 was a record year for algal blooms in Florida. It put water quality really at the center of policy and political issues at that time, because it was also an election year. And it's known that these harmful algal bloom or HAB events have a significant economic impact on many of our activities, commercial fisheries, public health, tourism, and trying to manage those blooms as well. But so this is pretty well known to have these significant economic impacts from these blooms. So we have a number of agencies in the Indian River Lagoon and the St. Lucie Estuary that are monitoring algae within our water bodies. South Florida Water Management District, DEP, they do a lot of the blue-green algae monitoring. And then some of our universities like Harbor Branch has sensors out there, and experts that regularly are monitoring for potential harmful algal blooms that might occur. And then FWC has a red tide offshore monitoring program that has been in operation for quite some time. Probably most of the focus is on the West Coast for that. But given that, the question I had, coming into this and starting as a Sea Grant agent, is there a role for the public to engage the public in being not only educated about harmful algal blooms and phytoplankton in general? Is this something that we can get the public involved in? And I had already known about the Phytoplankton Monitoring Network from working with it in my previous position. And it's a great program. It's a national program that is operated by the lab up in NOAA lab up in Charleston, started by Dr. Steve Morton up there. And the whole idea is to train citizen science to collect water samples. They train them to ID potentially harmful species. And then there is a national database where that information gets entered. And the data that citizen scientists or community scientists are collecting can then be used by not just NOAA scientists, but all scientists. And currently they have 3 programs: marine program, freshwater program, where they're really focused on cyanobacteria, and just recently launched an aquaculture program. And I'll describe a little bit of that as we go along. But what's cool about this program is that they really do a great job of training the public on not only how to sample the water, but also how to identify the main species of phytoplankton and cyanobacteria that are of interest. And so they recently, with the pandemic, they've done a lot more online training sessions. I've had my fellow Sea Grant agents participate in one last year to see if they can bring this program to their respective areas. But they would, that's Jennifer Moucher right there, she would actually travel to the different locations throughout the country to do in-person trainings. And that's actually when I first started, we got that set up and had a nice little cohort of volunteers that a few of them are still actually acting as PMN volunteers. And they have these great resources. They developed an ID guide for cyanobacteria, and I'll show it a little bit later, this app called Phyto that you can download on your phone, and it really helps, especially when you're out in the field. But the idea is pretty simple. You do a small plankton 3-minute tow. You collect water samples, analyze the samples that are collected through that tow net, that plankton net, and then after training, you get to the point where you can actually start identifying the species. And there's this whole QA/QC process that you go through with Jennifer or Nia to refine and hone your abilities. So if people are unsure whether or not they can do this, it's really they're very good about getting everybody up to speed and trained, and their initial data is QA'd and QC'd so that they get more comfortable. But this is kind of the data sheet that they use, and it's pretty broad categories as far as distinguishing the amount. We don't do cell counts. It's really like, is it present? Is it no, yes, and are there elevated levels of it? And in the training you do get trained on how to define what is elevated, certain percentage that's taking up the field of your microscope. And so it's and then you take basic water quality measurements, air temp, water temp, the salinity, those kinds of things. So, and what's really neat about this, the level of training that they provide, but they also supply all the materials to the volunteers. So a refractometer and thermometer, a plankton net, slides, and then all the ID sheets that people need for reference. And then if there is a particular, I might have glossed over that, you can see here, if there are particularly elevated levels of certain species that are of interest because they can be harmful if they're blooming, they do ask that people send a preserved sample of those so that they can truly verify and really get those to exact species if need be. So that's one of the great things is working with them as a Sea Grant agent and kind of recruiting volunteers to participate, and they do all the training. So I mentioned that Phyto app, which is a really cool way of learning on the fly as you're sampling, and they have it for both marine and freshwater species. And during the trainings itself, they're pretty fun because you get to learn all these different mnemonics to remember species. This is the diddlem or the Tootsie Roll, although I kind of remember the Tootsie Roll more than the actual species. But this is a common diatom that you might find. And there is a focus on harmful species, but they do still want people to learn other species that may not be harmful. And that can be particularly important for the aquaculture program of the PMN. Here is a dinoflagellate that is known to be toxic, Alexandrium species. Again, we're really getting them to just genus. But here's another way to remember this particular one. It looks like a stack of hamburgers. So we get to do fun things like that in our trainings. And the other thing I forgot to mention, and they used to do this, I'm not quite sure if they still do this, but at least in the freshwater program, at a certain number of tests that a volunteer would do, they get use of a microscope. So the NOAA PMN will actually send that individual microscope once they've sort of proven their worth, so to speak, in terms of the consistency of sampling. And then, of course, all their IDs being QA'd and QC'd. So as I mentioned, it is a national program. It's in 14 coastal states, including the Great Lakes as well. This is maybe not so up to date as a slide, but over 100 volunteers and over 130,000 data points submitted since 2001. And of course, it's as I mentioned earlier, a wide range of participants, school groups, just interested individuals, nonprofits, and local governments as well. I recall one training that Jennifer was doing in Texas. A lot of the local state agencies were actually taking that course so that they could actually use that data within their agency. And so some of the main benefits of this program is you can be a little bit more nimble. You've got this wider range. The agencies will have these set stations that they will sample at, but their blooms might start in other places. So it helps increase the geographic spread of knowing where blooms might originate and enables that sort of early warning system that this program could allow. And there's a number of different benefits that I show here, but really the one I like to highlight is really promoting that increased awareness and education to the public on harmful algal blooms, but also just phytoplankton in general. Not every species is harmful. One thing that we always point out in the trainings is that we would not exist without algae or phytoplankton, because those are the organisms millions, billions of years ago that created oxygen on the planet. So and in the oceans. So National impacts. Again, we're well past 20 years, but 21 years now. So this is a little out of date, but this is just showing some of the impacts that this program has had. Being able to identify and be that early warning system for important toxic events. And some of the examples of that data being used is some of these agencies are now using a lot of that PMN data to make informed decisions about what they should do, particularly when it comes to things like seafood safety, in some particularly bad domoic acid base blooms that they've had in the Pacific Northwest. So my role as a local coordinator is just trying to, again, recruit volunteers to get them interested in the program, facilitating those trainings with those individuals. But there's been some improvements in the program recently where that has they've improved the database so that I can have direct access to the data within my programmatic area. So they've the PMN has actually created a Florida Sea Grant project for not just for me, but all Sea Grant agents throughout the state, where if they're involved in that program, they can pull that data out and then look at if they have volunteers in their area, they can pull that data out and report back to the public, which is really what what I want to do, and what I think a lot of my colleagues want to have the ability to do is report out findings in a timely fashion, because these things can be so dynamic and ephemeral. And one way that I'm planning on using that is I'm part of a bi-weekly or bi-monthly harmful algal bloom conference call with local managers within the Indian River Lagoon. Thankfully, I could say that we haven't really detected any blooms with our volunteers at this point. Just occasionally presence of some of the more toxic ones. But after our initial training, we set up some sites here. The blue dots are where the PMN sites were initially set up this is in the St. Lucie River in Martin County. And then we also had a couple spots in St. Lucie County as well. A freshwater spot in a local nature preserve. And then also on the Indian River Lagoon, a tributary that runs into the lagoon, and then also directly on the lagoon. And these are all, especially this site here, most of these sites are of public interest. This site here is a park where there's a lot of people entering the water, swimming, boating, fishing. So, and it's also monitored by the health department for enterococcus bacteria as part of the Florida Healthy Beaches program. So there's I think a lot of synergy that can be accomplished with this program, and also getting the public and the community engaged in community science, particularly in an issue that really is of extreme interest and concern in my area. So with that, I'll end here. If you have any questions, I see I see some numbers in the chat, but I will try to copy and paste these emails here. Jennifer Moucher is the lead coordinator for the NOAA PMN program. Nia Renee is a new coordinator that has just started the aquaculture program of the Phytoplankton Monitoring Network. And I've had one of our fellow Sea Grant agents that's trying to get that started in the Panhandle. And the purpose of that program is to get the shellfish industry involved in understanding or knowing what particular species of phytoplankton might be, either whether it's on their farms, their nurseries, or their hatcheries, and especially if they're species that might either adversely impact their crops, their clams or oysters, and even to keep track of blooms that are beneficial for the shellfish growth. So I think the last thing I need to show here is we will put up the poll, and I will I can go through the questions here, or if anybody, Anna, are you supposed to read these off, or?
Yeah,
Oh,
I
thank
can
you
read.
for doing
There
that.
they are.
Okay, all right. Let me look over here. Okay. Oh, and thanks for posting those links there. I really appreciate that. Okay, here's from Laura. Have we seen any reduction in bloom size or occurrences in response to various mitigation efforts? Anything that works? Whew. Well, in the Lake Okeechobee and the St. Lucie River, I believe, I don't know if I don't think it was 2018. It might have been the year after, or maybe 2019. There was a company from Israel that was hired by the state. They had a particular technology that was that they had developed to treat blooms, particularly at the source. It was a chemical agent, peroxide based, and they did some test runs, but there wasn't, if I recall correctly, there wasn't really any acute blooms occurring at that time. So I think that the whole mitigation strategies are pretty controversial. I think can be controversial, and there's other things that people have tried, clay treatments, fine clay treatments to make that bloom, those bloom species of cells sink to the bottom, but that can have some potential adverse impacts as well as chemical treatments. Another thing, there is a company based in the Scandinavia, I believe, LG Sonic, that has developed a sonic technology that they can deploy off blooms, and apparently they're using that in reservoirs in different countries. I know that in the Southwest, they had tried, I think they had done some tests in some of the lake systems or pond systems out there, but I don't know the results of that. But the whole idea is that the sonication, the sonic energy that's emitted from that instrument will break up the cells. But then, of course, if you have toxic cells, you might kill the cells and release the toxin. And so that is also can still be a problem. I will say with the dogs, that the dog toxic events that happened in 2018, they also happened in areas where the bloom was not highly visible or noticeable at all. So certainly there was toxin in the water at that time.
Hey, Vincent, this is Anna. I'm gonna just politely interrupt you for just a second.
Mhm.
And I wanted to just acknowledge that it's 1 o'clock. So if you all have to end your lunch break or go back to work or walk the dog, please feel free to do so. We're gonna stop the recording, but we will be staying online to have question and answer with Vincent. He's been doing a wonderful job so far. And Vincent, I don't know if you had an ending slide, otherwise you can just unshare. So that way your viewers can interact with you.
I think that was it.